Hi there, this is Kristen Rossler. I'm going to go through the week 4. We're on week 4, right? Is it week 4? Yeah, I think we're on week 4 in terms of Bio 1011 labs. And then this one is, it's kind of the last one that's very challenging, I think, as an instructor. So just bear with me. We're going to get through this lab, and hopefully, and things usually get a lot easier after this week. So this is the setup. So each group is going to have 2 surface probes and one of these big tankard things in the middle with on a stir bar heated plate. And we're going to have 3 probes, and that's why we need the 2 different computers, because each computer can only accommodate 2 probes. So this is kind of the best way to set it up. So every once in a while we have a group or a lab section that's large, and that presents some specific problems. So if you have more than, I'd say, 4 groups, you may want to consider not doing one of the probes. And the probe that I would get rid of is actually the oxygen probe. And by getting rid of the oxygen probe, you will have, of course, more computers and more SparkLink portals. Sometimes it becomes an issue, right? So if you're having, sometimes just the computers, especially if you're a group that's in the afternoon, the computers have lost their charge from the morning group. Sometimes we just don't have enough of the SparkLinks that are functional, or the probes that are functional, etc. So one thing that you could get rid of, and if you wanted to, is the oxygen probe. And to get rid of that, you can always just keep the probe on. Like, see here, the setup that I've got here. The oxygen probe is the one that's closest to us, and you could just unplug it, right? And so then not actually have it. So it still has a gasket in there that's going to keep, the conditions inside, in anaerobic, conditions. So that's just my 2 cents. But if you do have just 4 groups, go for it. You can have all 3 of the probes, and this is kind of what the setup's going to look like. The way that I have them do it, and I think it works pretty well, is that on one of the computers they would have just the probe for oxygen. So I have that over on the right here in this example. So I have my oxygen probe, which is closest to me, that's hooked up to the computer on the right, and then the other 2 probes, the carbon dioxide and ethanol sensors, those are the ones that are actually going to be revealing the products of anaerobic respiration. I have them hooked up on the left. So that's the way I do it, because I want them to remind them that the one on the right, the oxygen sensor, is just proof of concept, right? So we're hopefully going to see a dip in the oxygen levels that just proves that we're getting into anaerobic conditions. And whereas the one on the left with the ethanol and carbon dioxide is really generating the products. And obviously we would not get any ethanol being produced at all if it weren't in anaerobic conditions. So anyway, so I think the oxygen probe is sometimes a little bit repetitive and not quite as necessary. So if you want to get rid of one and just make your life a little easier, or if you don't have enough computers, like I said, or the computers aren't charging, etc., probes, just get rid of the oxygen one. Anyway, here we go. So either scenario, you're going to set it up very similarly to how we did it last time. So here we're going to have, let's just kind of, so each group, let me just show you on the board here. I went ahead and drew a picture of the basic setup, right? So if you are doing all 3 probes, they're going to have the thin ethanol probe. It's going to be reading in percentages. That's the units, of course, that you want to make sure the ethanol, I think it's automatic, but you just want to make sure it's on that. We're also going to have a carbon dioxide probe that's in parts per million, and then the oxygen probe here is in parts per million as well. Each time they do a setup, they're going to do at least 3 setups. They're going to have 1 liter of yeast that's been prepared by Tracy, or the lab tech. And the dilution is, I think, described in the packet of information. Can't remember it off the top of my head. And then you're always going to be, so they're going to have their liter of yeast with their stir each time, and then they're going to add to it 500 mils of a sugar-water combination. So here's the sugar-water combinations that they can use if you want. You can, really kind of use whatever you feel like is necessary. I think Tracy has accommodated for these 3 molarities, so that at least in our lab group we have enough sugar. We should have enough sugar for all of these combinations for our lab groups this week. And so the first one, 0.25, is actually kind of a hypotonic concentration, obviously, of the sugar. and we're going to see that it has some impaired, effects on the cell. As we get closer up to, I think it's around 0.3, 5-ish, you're going to start seeing that it's more isotonic, and then greater than 0.75, we're going to start seeing hypertonic, right? So it's kind of fun to see these three different osmolarities and how they're kind of affecting the rate of anaerobic respiration amongst these yeast. So each group is going to do one specific sugar if you have 4 groups, right? So what I typically do is I have group 1 do dextrose, reminding them that dextrose is just glucose, right? So it's a monosaccharide. I have group 2 doing another monosaccharide, fructose. And then I have group 3 doing, what do we have group 3 doing? Oh, we have group 3 doing usually lactose, and then group 4 doing sucrose. You can do whichever combination you want, all right? So, but they have to get through all 4 sugars. So there'll be 2 monosaccharides and 2 disaccharides, obviously, that they'll use. What you're going to see, of course, is that it takes longer for the disaccharides to be utilized by the cells. And the cells have some preference, of course, for the different sugar substrates. I tell them it's just kind of like humans. We have different preferences in terms of what we like to eat more. I like tacos more than salad, so I tend to gravitate towards that first. That's always a cute little example. We talk about how to do the concentrations. So I usually erase this and have them work it out, and then check every group's work. But here, of course, is what it should look like. It's just using that M1V1 calculation equals M2V2. Tracy has made 3 molarity concentrations of each of the sugar substrates. You're going to see that the lactose usually looks kind of brown. It still works fine, actually. We've tested it out. It just kind of gets oxidized, I think, and it seems to be working quite well. So the students are alarmed, don't be alarmed. And I think there's another one that's sometimes yellow. Again, it's all good. So I just did the calculation here, obviously, for a 0.1 molar final concentration, just because it helps me do the math fast. And of course, that would be 50 milliliters of a total, kind of solution for a 0.1 molar solution. So just extrapolating that, a 0.25 molar sugar concentration, I'd have to use 125 milliliters, right? And so forth and so on. So I think you guys all know that. So I have them typically do at least 3 runs. If you're feeling bold and there's a lot of sugar around, maybe do the 4th. Maybe hold off on the 4th just because we need to make sure there's enough sugar for all the different lab sections. But anyway. So they're going to do 3 different runs, each for 20 minutes. All right, and hopefully that's all making sense so far. I would encourage you to put a picture up like this if you don't have one for the setup. So let's look at how it's going to work. So just to remind you how these computers work, so doing the oxygen, it's pretty straightforward, right? So just the one graph that the students are going to generate, and the big thing is that you want to go around and make sure it's on parts per million and not percentages in terms of the units of oxygen. It's a pretty easy thing. They're going to get everything set up like so, making sure that the holes have the gaskets in them and that there's no, obviously, oxygen getting in or out. I like to have it oscillating in at about 35 degrees or so, just to keep the yeast kind of happy. For the other graph, just to remind you, since they need 2 different sets of data, let me just remind you how you could set that up. So let me see if I've got this on the computer right. So we're going to build a new experiment. And here, if they build a new experiment, they have a bunch of different options. We want to go with the side-by-side because they're going to be doing 2 sets of data. So you can see it's over on the right side, about halfway up. And then there's 2 kind of equal-spaced, graph areas. All right, and so now we've got, and you can, have them set up. One is ethanol and one is carbon dioxide. Again, they would just hit for each of these, they hit the graph. And then they're going to have to select their units over here in the left kind of thingamajigger. So we're going to say the left one, how about, is carbon dioxide concentration, and make sure it's on parts per million. And of course, for this one, we're going to do ethanol and make sure it's in percentages. All right, and then they can change the time if they want, down on the bottom. There's on the x-axis, of course, there's these little times. I think it comes, the default is in seconds. But of course, they could change it to minutes. There it is. All right, I think I got that all changed correctly. And so now they're just going to run, once they have everything set up properly, they're just going to do the same thing that they've been doing for the previous labs. They're just going to hit the start and let it run for 20 minutes. All right, so just FYI, some things that you may or may not have realized about how to do the calculations. They're very confused on this lab in terms of minimum and maximum. So if you look at the template that I made for it, I tried to make it very understandable that oxygen is going to drop. So the maximum is our initial read, right? And the final is the minimum, right? So we're going to see a drop, of course, in the oxygen concentrations. So maximum is initial, minimum is final. Conversely, for the oxygen, or for the carbon dioxide and the ethanol over here, they should see, of course, a big growth, and they do, usually works really well. And so, of course, the minimum would be the initial, and the maximum would be the final. You'd be amazed. It takes oftentimes a fair number of times of explaining it for the students to remember that. So they're going to have to record minimum and maximum each time the same way that they did before. Another thing that you can do is that if the students, sometimes they mess up in terms of how they've been, setting something up, you can always drag on specific units. Where's my mouse? Let's see. So if you look down here, I don't know if you can see it. There is the default down here is where is it? I wish I could have had it. Oh, do I have a? Let me find my pen. Here we go. So the default down here on the left is just for the mouse. You can actually select the box right next to it, just right there, that gray one right there, and it allows you to toggle and select on specific data points. So for instance, sometimes students will turn it on prematurely and does these erratic things at the beginning, and if you wanted to get rid of all that, you would just use the box to actually select for the material you want. So I'm just going to do that real fast.